Stuart, and good morning everyone. It's so wonderful to be here this morning for the NRF Brain May event, and it's fantastic to see such a great turnout this year. So, yeah, we're shifting from glioblastoma to medulloblastoma. So just a bit of background on medulloblastoma. It is the most common malignant malignant brain tumour in children, with a median age of diagnosis from around 5 to 7 years. But it does occur in infants, and it can also occur in adults, although it's quite rare in adults. Now, these tumours grow in the cerebellum, so that's this region of the brain here that I've circled in red. So the cerebellum's at the back of your brain, and it's a really important part of the brain, so it's responsible for movement and balance. So children who develop medulloblastoma will often present with abnormal gait or movement issues, tremors, clumsiness, etc. So there are around 25 new cases of medulloblastoma diagnosed in Australia every year in children under the age of 15. And current treatment consists of surgery, so to remove as much of this tumour as possible, and this is followed by chemotherapy and radiotherapy. Now, whilst these treatments are quite effective at controlling the tumour, they are very toxic, and we have to remember these are brains from children, so they're developing brains. So these treatments do lead to long-term side effects. Therefore, we need to develop more effective and less toxic therapies, which is what we're trying to do. Okay, so not all medulloblastoma tumours are the same, and we can stratify these tumours based on their molecular or their genetic profile. And for medulloblastoma, there are 4 distinct subtypes. So we have WNT, Group 3 and Group 4. I'm not going to talk about these subtypes today. I just want to focus on the sonic hedgehog subtype of medulloblastoma. So this is a common subtype. It accounts for about 30% of all medulloblastoma cases, and it is named sonic hedgehog after the gene that is hyperactivated in these tumours. And just a fun fact, the sonic hedgehog gene is actually named after the video game character from the 90s, Sonic the Hedgehog. So this is just a cross-section of the cerebellum. So in normal cerebellum development, sonic hedgehog is very important. It instructs immature or baby neurons to grow and divide and to move to the cerebellum to where they are needed. Once they are there, the sonic hedgehog pathway gets switched off and these cells stop dividing. However, in medulloblastoma, there's mutations in certain genes in this sonic hedgehog pathway. So this sonic hedgehog is hyperactivated. It never gets switched off. So these neurons never get the signal to stop dividing, so they just keep growing, and this forms a tumour. Okay, so we've been looking for new therapeutic targets for medulloblastoma. And one of the proteins, which is actually an enzyme that we work on in the lab, is called sphingosine kinase 1. And I'm just going to call it SK1 for the rest of today's talk. But when we looked at the expression of this SK1 gene in medulloblastoma tumours, what we found is if we look at the 4 different subtypes, that specifically in this sonic hedgehog subtype, which is represented by this blue bar down here, the SK1 gene is really highly expressed. And to look a bit more deeper at this, if we analyse patient survival data, the patients that have high level of SK1, so that's this blue line here, have a poorer survival. So this led us to ask the question, do high levels of SK1 contribute to the pathogenesis of sonic hedgehog medulloblastoma? So let's just look a little bit more closely at what this SK1 protein does. So SK1 is involved in a pathway which metabolises a special class of fats which are called sphingolipids. Now, sphingolipids are really important components of our cells. But basically, SK1 can convert this fat molecule called sphingosine into this molecule called S1P. Now, what's important about these molecules is that inside the cell, sphingosine can cause a cell to die. However, this molecule S1P is needed for cells to grow. So it's sort of like a bit of a balancing act inside the cell. If we have more of this S1P molecule, the cell will grow and divide. However, if we have more of this sphingosine molecule, the cell will likely die. So SK1 really controls this balance between cell death and cell growth. So what relevance does this have to medulloblastoma? Well, as I told you previously, we know in sonic hedgehog medulloblastoma there are really high levels of this SK1 gene. So this leads us to assume that there is also high levels of this cell growth promoting S1P molecule. So this can be contributing to the very aggressive nature of these tumours by making these cells grow more. So this leads to our hypothesis that targeting SK1 could be therapeutically beneficial in sonic hedgehog medulloblastoma. So how we are going about testing this in the lab is by working with some sonic hedgehog medulloblastoma cells. So these are cells that were taken from an actual patient tumour, so a patient who had sonic hedgehog medulloblastoma, so this was many years ago, and we can grow these cells indefinitely in the lab. And if you're joining one of our lab tours later, you will probably actually get to physically see these cells up in the labs. So what we can also do is do gene editing of these cells. So we can cut out or delete that SK1 gene from these cells, and then we can see what effect removing SK1 has on the growth and the survival and also the migration of these cells. So in the interest of time, I'm just going to show you one piece of data that I'm particularly excited about, and this is cell migration. So to do this, we plate out our cells, and then we create a scratch or a wound in the cells. And then over time, we can measure how these cells migrate into that area. So you can see here our control cells, which have high levels of SK1. When we create this scratch, as you can see by these black lines, after 30 hours, all of the cells have migrated into that area. So these cells are migrating really fast. However, when we delete the SK1 gene, after 30 hours, we still have that scratch present, and we can quantify this. So this data tells us that SK1 is playing a role in how sonic hedgehog medulloblastoma cells migrate. And how tumour cells migrate is really important because this is how tumour cells can invade into our normal brain. Alright, so I'm just going to finish up now on some work that we've been doing with SK1 inhibitors. So we have compounds in the lab that can block SK1. So these are light drugs, but they're not in the clinic yet, so they're not clinically used. But what we can do is we can take these SK1 inhibitors and we can treat our medulloblastoma cells, and then we can see what effect that has on the growth of the cells. So we've done this. We've used 2 different SK1 inhibitors where we have treated our sonic hedgehog medulloblastoma cells. And this line here represents the viability or the survival of the cells. So as we add increasing concentrations of SK1, these cells are dying. So these SK1 inhibitors are really killing these cells, which is very exciting. And I think what's also really important is that we know that we have mice which have no SK1 protein. So mice that have had the SK1 protein deleted. And these mice are normal, healthy, they have normal brains. So we think that this therapy is not going to affect normal, healthy cells. Also, when we have used our SK1 inhibitors in mice, these inhibitors are really well tolerated, so we don't really see any toxic effects on the mice. So this is telling us that maybe SK1 is a good target for medulloblastoma, and inhibiting it might lead to those less toxic effects for the children. Alright, so just to summarise, we've identified SK1 as a promising target in sonic hedgehog medulloblastoma. Deleting SK1 in these cells reduces their capacity to migrate, and also SK1 inhibitors really effectively kill these sonic hedgehog medulloblastoma cells, and also these inhibitors are well tolerated in mouse studies. So on that note, I would just like to acknowledge all of the people in the lab who help with this work. So Professor Stuart Pitson, who's our laboratory head, and all of the Brain Tumour Therapeutics Group, who is co-headed by Dr. Melinda T, who is sitting down the front here. All of our RAs and students, most of whom are here today. I would really love to thank the patients and the families. The patients and their families, their stories, they really motivate us. Research can be a real slog. So hearing these stories just makes us want to get up and go into the lab each day and work harder and do better. I sincerely thank the NRF and the amazing donors, the NRF donors. Your support just means so much. It means we can do this research. And particularly Munno Para Foodland, who sponsored the research that I've shown you today. So thank you for listening.
Any questions for Briony? I guess the obvious question that comes to mind is that there's promising data there. How long do you think it's going to take to, if it all works well, to be able to translate this into a new therapy for patients?
I wouldn't like to put a timeline on it, but there is still, so this is a very new project, we've still got a lot of work to do, but I suppose where we're moving to now is really, as Nicole said, we're taking this into more clinically relevant models. So we've got a lot of mouse models of medulloblastoma. So what we would like to do, first of all, is put our cells where we've deleted that SK1 into mice and see how these cells actually grow as a tumour in mice. And then try treating, have mice that have medulloblastoma tumours and try treating them with our SK1 inhibitors and see if we can actually, delay the growth or even cure the mice. So they're the next steps. I think, a trial is probably still quite some time away, but.
And I guess the other point is, we're very fortunate, that we have patients that are generous to be able to allow us to do research on the tumours, and with help of the next speaker, Jordan Hansford, we've been able to access patient material from children who have gone through the regular surgery and be able to then play around with that tumour material and see whether with a real tumour material rather than some of these cells that were isolated many years ago, whether the effects are still there. I think we're seeing those effects. All right, in the interest of time, we'll move on. So thank you, Briony.